right. Now we have the final portion of the conference for today, our Q&A. So we're going to call up all of our student speakers for the day. So just give us a minute to get them all on our screen. There's a Stephen here. I'm guessing that there's no last name, but okay. Great. Well, it looks like we have all 6 of our speakers here with us. Awesome. So we have several questions for all of y'all. First of all, I know a lot of you are coming to us from the UK or from Europe, so thank you for being with us so late. I know it's quite late there, so I really appreciate that. So we'll just go through each speaker in turn and ask the questions that way. So I'll just start with Javier. You're first on my screen. So the first question is, what methods could be used for further research if DMT is stored in synaptic vesicles?
Hi, everyone. So the question is, how could we check that, isn't it?
Yes.
Okay. Well, we could do it either in vitro, for example, using synaptosomes. We could use radio-labeled DMT to see if it's uptaken by synaptosomes, and then using different drugs like BMAT2 blockers to see if there's a difference between the distribution. And another way would be in vivo with a microdialysis approach. We could monitorize DMT levels in some brain region of interest, and then using, for example, reserpine to deplete monoaminergic synaptic stores and see if there's any difference in extracellular DMT levels, or, for example, with potassium to force synaptic release by depolarizing the neurons.
Okay. Thank you. Thomas, I have is next. So this question is, you mentioned there should be little to no modification to sensations or reflexes with these synthetic psychedelics. But what about other aspects of the mystical experience that many researchers think is the key to psychedelics' clinical effects? How much how much can or should be changed about the natural psychedelic experience?
I don't know if I got the question. I mean, if there could be some relationship between endogenous DMT, for example, and some other kind of mystical experience, not near-death experiences. Is that the question?
Well, I think this one is aimed for Thomas.
Yeah.
So this is
okay.
Thomas. Yeah.
Yeah, it's a really interesting question. I think that very mild subjective effects and mild sensory impairment is interesting, A, because it makes these drugs more likely candidates for daily use. Say there's some ibogaine derivative that has no change in sensory perception but still carries anti-addictive effects. That's a really interesting clinical outcome. I also think that the tie between the intensity of the acute subjective effects and the long-term impact of the psychedelic is pretty well substantiated. Like if you look at a lot of these critical period research papers, the longer you're tripping, the more socially sensitive you are after that trip. I think there's an element of like intensity of that as well. The main reason I advocate for not significant sensory enhancement, is that I think you can have really powerful internal experiences that might not come with perhaps some of the cultural jarriness that like classic psychedelic trip has, and that jarriness and that somewhat incompatibility between significant sensory enhancement and, oh, well, double blinding standards that have just taken MDMA significantly back as a candidate at the FDA. Like, we have to adapt to the moment in the culture. And I think the moment in the culture is looking for something that is definitely milder than MDMA and potentially milder than psilocybin, so that these experiences can be scaled and perpetuated in a way that isn't too socially jarring. And also, I think that intense psychedelic experiences are fundamentally key, and that the most responsible thing we can do is adapt cultural containers for classic psychedelic experiences, and that this mild psychedelic would just be a step towards that.
Got it. All right, thank you. Next, I have Stephen. What do you believe are the ethical implications of using AI to try to classify human experience? Do you believe this would oversimplify observational research?
I guess it relates to what I was saying during the during the talk about transparency. So it's about the engagement of the researcher with the research process. So if we have an AI that assists in the classification, there should always be a human that stands at least for the validation of the classification, so that with their name and with their, let's say, authority, they say, okay, this is how I would classify, and we just automated this process through AI. Surely there's something lost. There's also just something lost by not talking to people, but just looking at the words. So the tone of the voice, facial expression, all that information, it gets lost if you only have a language model that looks at statistical relations between words. So there's already so much that's getting lost. But I would argue, if you have to compare it to the status quo of research right now, and that is giving questionnaires to people mostly, where they convert basically text prompts into numbers, and then it gets transferred into a statistical analysis program. So I would argue that the AI classification process I'm proposing, if it's done transparently and consciously, is probably more embodied than what we're doing now.
Oh, okay. Thank you. Next question is for Sabi. Can you speak on the possible implication for individuals with dysregulations or disruptions in their mirror neuron systems?
So meaning if they take psychedelics, what would happen? Well, if there is a down regulation, then I would predict that the mirror neuron system would not be able to engage as much, and therefore this connectedness aspect of the experience and empathy aspect of the experience might not be that prominent in that case. In case of an upregulation, then obviously the opposite would be predicted.
That makes sense. This question is for Marcus. Can you speak on any implications of DMT being short-acting versus psilocybin being long-acting, and if this affects the efficacy of each? Marcus, are you there?
Hello, yes, I am there. Please, can you repeat the question?
Okay. Yeah, no problem. Can you speak on any implications of DMT being short-acting versus psilocybin being long-acting, and if this affects the efficacy of each?
Yeah, it's a very good question. So in the context of our specific intervention, we use this whole memory reconsolidation approach. The time window in which that takes place is between 10 minutes and 6 hours after reactivation. So we need a short-acting psychedelic to act in that time. Obviously, there's DMT, which is 20, 25 minutes, which is perfect. It fits in that window. Psilocybin extends beyond that. It gets a bit more confusing. So in the context of memory reconsolidation research, which can be applied to addiction, PTSD, and phobias, DMT works very well as an option. Regarding other disorders, I'm not entirely sure. It does seem to be the case that both DMT and psilocybin have efficacy in the context of depression. Whether there's a difference may be down to their difference in duration or maybe down to other factors. It's currently unclear.
Okay, thank you. Now we have a question for Victor. What are the biggest opportunities for AI and brain-computer interfaces to disrupt psychedelic therapies? Drug discovery, real-time brain monitoring and modulation, understanding mechanisms of action and drug interactions, personalized psychiatry, accelerating clinical trials, or something else?
Well, in advance, I want to say that I'm recovering from a cold, so I apologize for my voice. And secondly, I appreciate for everyone taking the time to watch my talk. So in regard to the question, something that I want to clarify and that I want to say is that utilizing AI research methods into neuroscience to be applied for new therapy is not supposed to replace human interaction and is not supposed to replace the responses that we see after a psychedelic treatment. With that being said, that the introduction of AI into this type of research, it's only a tool that would help us validate current information and that will help us get more information into how to create better treatment. So that's a very important point. So I think that's one of the main opportunities that we can take into this, because we can use this tool alongside other research techniques that we can use to create new therapies. And once again, there's new therapies in order to help people, but I think it, as they mentioned in the question, is very interesting how we can maybe utilize this for brain synchronization, or maybe we can utilize this to create brain interfaces that will tell us more in a real-time experience with a higher resolution what's happening in the brain during the psychedelic experience.
Got it. Thank you. All right, we'll go back through starting from the top. So Javier, can you speak more on DMT versus serotonin on the 5-HT2A receptor? What would be the point of us having an endopsychedelic subsystem? Please speak more about the endopsychedelic subsystem and its significance in general.
Well, that question is huge. I'm very speculative because it's an hypothesis. I mean, I don't know if even it exists. I just propose it. However, I can speak about differences between 5-HT2 activation between DMT and serotonin. Actually, it's very different. The response at several levels. You've got different electrophysiological responses with, for example, psychedelics activating 5-HT2A receptors depolarizes the neuron in a peculiar way with longer postsynaptic excitatory potentials that you get with serotonin. Also, you've got a recruitment of different signaling cascades. This is called biased agonism, and it's important for psychedelic to understand psychedelic action. Not every ligand binding to a GPCR, a G-couple protein receptor, activates the same pathways, intracellular pathways, with the same efficacy and potency. So you've got also differences in these downstream cellular signaling cascades that are different between psychedelics and also within psychedelics and serotonin. And also you've got this ultimately leads to differential gene expression. For example, both serotonin and both hallucinogenic and non-hallucinogenic 5-HT2A receptors elicit CFOS expression, for example. But there are some reports, few, but there are some, that only hallucinogens, for example, elicit the expression of EGR1 and EGR2 genes, which are early growth response genes that are have been related to plasticity, for example. Also, we've got this effect. The serotonin is unable to induce plasticity in neurons, while psychedelics do. So we've got at many levels, like electrophysiological level, cellular biology levels, and also genetic expression levels, different biological meanings, so to speak, of the same receptor being activated that are different between psychedelics and non-psychedelics. I mean, that the point is, if you could inject serotonin inside your brain, because serotonin does not cross the blood-brain barrier, so you would have to inject it directly. Probably you wouldn't trip. Maybe you would die. You would have a lot of nausea and headache and agitation. You wouldn't have a trip. I mean, it's. Well, there are some. My opinion is that it's mostly about a bias activation of the receptor. There is also this proposal of the activation of intracellular receptors. I don't know. It's a complex issue, this one.
Yeah, of course. Complex issue. Okay, thank you. Thomas. So other than the work that the Shulgins are doing, or I know she recently passed away, but what work is recently being done in producing new recreational drugs? So of course, there's many ethical implications for doing this research in publicly funded labs. Do you know what work is being done in that arena?
Yeah, I know that the Sames lab in Colombia is currently pursuing this. I know that the Olson lab in Davis is currently pursuing this topic. I know that the WIPF lab at the University of Pittsburgh is. You have some private companies such as Tactogen that are very directly targeting milder MDMA derivatives as a means. Mainly they're speaking about cardiotoxicity, but like consent issues and enthusiasm are like the scary thing under the table that no one quite wants to say out loud. And what else? I know Mindstate Design Labs is working on some of its own chemicals, and the Alexander Shulgin Research Institute has taken over a lot of Shulgin's previous intellectual property and is developing it. Those are only the ones that I'm aware of. There are a lot of companies doing a lot of different things. There's a lot of companies trying to get their own flavor of psilocybin approved, which is a funny thing. But yeah, those are the places that I've been keeping my eyes on. I'm sure there are others.
Okay, cool.
Yeah.
Thank you for that. Stephen. So this is a follow-up from the previous question. What do you think of the ethical implications of using lots of data-driven features of people's transcripts to analyze how intelligently and/or organized their reports are, or to decide how mentally stable they are after a trip? Where are we to place empathy or perspective, taking of a person that we readily call disorganized or clueless without careful understanding of their words, and for whom an AI classification software would agree with our quick judgments?
Okay, there's multiple things in there. So let's suppose we have a research question that looks at people's verbalizing ability. That might actually be something interesting to look at, since there's this factor also in IQ measurements called verbal fluency, and people have differing abilities on this factor. So in research in general, this might be a whole like big confounding factor, that just people have different abilities to verbalize their experiences, and some people may be very good at that, and therefore their data might get, I don't know, overrepresented, for example, in research, while others might have experiences that are just as profound, but they just are not as good with words. Maybe they are very good with painting, but we don't analyze paintings, we analyze words, right? Mm-hmm. So Yeah, that's definitely something to look at. However, we have to keep in mind that we're doing research and not diagnosing people or telling people they are a certain way. So to go from analyzing the transcripts to calling somebody something or to say they're disorganized or whatever is quite a leap that you shouldn't do anyways. So yeah, those are my thoughts on it.
For sure, yeah. Thank you. All right. Question for Zabi. Has neural synchrony been studied in rodent models where you can achieve both high temporal and spatial resolution? So for example, in calcium imaging experiments, unlike can be achieved in human studies.
Actually, I think you got me on that. I am not really familiar with any animal model research in synchrony. So I think the whole idea there is to make a very naturalistic setting where people are freely interacting with each other. So I'm not sure how this can be translated to animal models. It is also unclear whether animal models like the rat have the capability for synchrony. And I'm not exactly sure whether mirror neurons are even discovered in rats. They were shown in macaques and also in humans. So it may not be there already at in the rat or mouse.
Okay, yeah, that makes sense. Thank you. Marcus, let's see. Would you be able to speak more regarding the right conditions for memory reconsolidation that would make DMT effective as a therapeutic?
Sure. So what we know about memory reconsolidation so far is that it works. And the process of memory reconsolidation interference is facilitated by other drugs. So nitrous oxide and ketamine and propranolol and a few other drugs. Whether DMT and other classical psychedelics can interfere with this process is something we're yet to figure out. It's something we're yet to test. It's quite a bold idea. This hasn't been done yet, but it has been done in rats in 2 different studies. So we suspect it will work in humans. Regarding what factors will affect this, firstly, we need to guarantee successful memory destabilization. So that's when you reactivate a memory and it becomes subject to change. It enters this labile state and becomes changeable. We have a paradigm that does that, and we've implemented that successfully alongside ketamine. That has worked. Yeah, it really does depend on. We don't know whether this will work yet. This has not. We have not done a human psychedelic trial involving this approach of memory reconsolidation yet. So it's really hard to say what factors will affect this. We're going to recruit 120 people onto our trial. So hopefully by the end of that, we'll have some data on what factors facilitate this process. But yeah, that's all I can say at the moment.
Okay, great. Thank you. Okay, and then back to Victor. Let's see. How might integrating AI affect FDA approval of the substances themselves, if at all? Considering it may be difficult to get the FDA to approve the drug in conjunction with another treatment, aka the AI technology.
Do you mind repeating the question again? Sorry.
Yeah, how might integrating AI affect FDA approval of the substances themselves? So I guess of psychedelics. So considering it may be difficult to get the FDA to approve a psychedelic drug in conjunction with another treatment, the AI technology.
Well, unfortunately, if psychedelic treatment is not approved by the FDA, while this research technique can still be used in neuroscience, as it had been used in other things, it can help push the field. However, I will be thinking that maybe using this in clinical trials or maybe in preclinical trials can maybe help corroborate and to gather and collect more information for a future approval. So it can be a potential tool that can be used to gather and collect more data that can be utilized for a potential approval in the future. But I'm not fully versed, to be quite honest, on the legal ramifications on how that would look like, and if it's even possible through the FDA clinical trial process.
Got it. Okay. Okay, someone put this question in the chat and said that anyone can answer. So if anyone knows the answer to this, please speak up. Are there any studies on psychedelics for pure hebephoretic presentations, or is this population not studied? And I actually had to look up that word because I don't know what it means. It's a type of schizophrenia typified by shallow and inappropriate emotional responses. If whoever asked this,
I'd like to make a comment on that.
please go ahead.
So I took the liberty to look it up, and as of now, I haven't found anything. And I'm not sure if I was clear or explicit in my talk, but I mentioned how a lot of the population picked for many of the psychedelic trials are only healthy people. And one of the reasons of the psychedelic action on people with schizophrenia, especially this type of schizophrenia, is that the saturation of the 5-HT2A receptors actually mimic some of the symptoms found in schizophrenic people. So people that are predisposed to schizophrenia through family predisposition or in other ways are normally not picked in the trial process for the same reason, because it can elucidate, of course, the schizophrenic symptoms. And as I said earlier, I looked it up and I haven't found anything that shows a lab or people doing studies on this type of schizophrenia and psychedelics.
If I may quickly, I know that Haley Duron is involved in this area of research, but it's a very small area of research. I don't know how much she's published at this point, but if you go into like Google Scholar and type Haley Duron, you'll probably find some articles like in or around this topic.
Yeah, that's a good point. And Chantal, you mentioned it's more disorganized thinking than straight up schizophrenia. So I don't know if you want to clarify more about this. Feel free to raise your hand if you'd like to talk about it. But okay, here we go.
Schizophrenia is a really unfortunate label. I think it's led to a lot of unfortunate exclusion of people who might actually benefit. I think there's obviously a lot of different subtypes that are all lumped together as basically incoherent. And I'm not fully sure if the type that involves basically no hallucinations, generally no delusional thinking, but a very fragmented sort of inner dialogue at all times with difficulty sorting out one's thoughts, necessarily involves a 5-HT2A receptor kind of chronic stimulation. And I think it'll be interesting to see like how that population responds to psychedelics. I guess it's just not really studied. So I just was curious because it's something that comes to my mind a lot, especially because I know that cannabis for some people can induce that sort of phenotype. Yeah, so I would just be curious if people who, have been adversely affected by cannabis would benefit from psychedelics, or if their brain actually might turn the experience into a kind of psychotic and like more adverse experience. But obviously, these are risky questions. They're just not explored. So that's all. I'm just curious about that.
Yeah, for sure. And you might want to also ask that question tomorrow for our psychology and clinical applications speakers. They may know a little bit more about that as well, but it's a good question.
Cool.
Okay. We have another question for everybody. Anybody who would like to answer? Has anyone studied psychedelics' potential to treat chronic pain? If so, can you speak to the compounds with the most promise?
There is one researcher at Maastricht University called Mauro Cavara, and they're currently they're running a study with psilocybin for fibromyalgia. So yeah, I actually conducted a podcast / interview with him. It's not released yet. The research department is sort of holding on to it. But yeah, if you connect with me on LinkedIn, I will send it to you as soon as it's available.
Cool, thank you.
Also, I am aware of Dr. Nichols from the LSU New Orleans Medical School. He studies psychedelics and specifically 5-HT2A receptors in the periphery in the body. And I know they're looking at responses in the mechanism of just psychedelics throughout the body and maybe finding a way to create new drugs that don't have the actual psychedelic hallucination experience. And at the same time, it's known that the 5-HT2A receptor agonists are very potent anti-inflammatory agents. So maybe that will be some cool research to look at.
Great, thank you.
I'd also like to add that there's also some research being done with sigma-1 receptor, which is it's not actually a receptor, it's kind of a chaperone inside the cell that has been studied for chronic pain and has a lot to do with cellular homeostasis and stress. And as far as I know, maybe I'm wrong with this, I don't really know, but I think that only tryptamines are activators of sigma-1 receptor. I don't know if LSD, for example, can activate it. So I guess that, but the issue here is that apparently you need very high doses to activate sigma-1 receptor. But this would make like tryptamines especially interesting. I guess, like, I mean, I'm not sure, 100% sure of what I'm saying, but I would say that tryptamines maybe can be more interesting for treating chronic pain because of this possibly sigma-1 mediated therapeutic effect.
Cool, okay, thank you. All right, I think we have time for one more question, so I'll just ask one general question for everyone, and we can go in order again. So if you could design your ideal experiment, money and funding is not an option, what would be this experiment? And it looks like the order has changed a little bit on my screen. Thomas, would you like to go first?
Could I have more time to think about it, actually?
Sure. The next person is Stephen.
So actually, I got really interested in this whole observational research thing because I think it's a great way to save money instead of running clinical trials. So I would just want to have a nice digital platform where people can connect to and talk about their experiences, and then have them be able to also verify the analyses that are being conducted with their data, so that they're actually seeing how the researchers interpret their transcripts, so to say. So yeah, however much money it takes to build this, and then to connect with retreat centers in different countries who are speaking different languages, and then comparing the languages and the narration between, like, I don't know, Japanese trips and Brazilian ayahuasca experiences and all of that. So we have a worldwide observational study, and I'm happy. So if there's anyone with this kind of funding watching, contact me.
Cool, sounds good. Next we have Sabi.
Yeah, so great question. Right now, I'm really deep into this topic of social cognition, so I will go along this line. And actually, I heard a recent talk from Thomas Palenicek, I believe, who is doing some hyperscanning experiments in the Amazon rainforest with shamans and just a very, very naturalistic setting, and measuring this activity in an actual ceremony. So I think this line of research would be very, very interesting, because mainly I think because shamans seem to have some kind of well, shamans who know their trade well, they seem to have a kind of an eye for selecting people, and also for managing these ceremonies, which may be seen as some kind of a social influence during the trip. And then seeing these social connections on a neural level would basically could underlie this effect. So I'm definitely very interested in kind of these social effects in a naturalistic setting. And you can imagine that we need those infinite resources and money to be able to do that, because it's a very, very messy area. It's very hard to capture any good signal. So yeah, it would be a great opportunity.
Sounds good. Next I have Javier.
Well, thanks for the question. If I were a rich man, which I'm not, I would first I would try to replicate these findings of comparable DMT levels to DMT levels comparable to those of major monoamines in rodent cortex, and I would check also other areas of interest like prefrontal cortex, of course, also limbic cortex, for example, and I would profit to do what I already mentioned before. That is assessing the differences in extracellular levels after applying treatments with vesicle synaptic vesicle depleters like reserpine, or and also with potassium to see what happens when you depolarize the cells. That's the first thing that I would do.
Cool. Yeah. Okay, Marcus.
Great. So the question was without any financial considerations, is that right? What about ethical considerations? Can I assume no ethical considerations?
That's up to you as the experimenter, how much of an ethical burden you want to take on.
Okay.
Please don't make any more TED
The answer I have in mind is without financial or ethical considerations. So I'll go with that. I'm thinking I'd like to see more on how sociological factors affect the psychedelic experience and psychedelic efficacy in clinical trials. So in my mind, a good study, a good way of testing this, an impossible way, but a good way would be 1 million genetically identical children or babies raised in all sorts of different scenarios, and then put through a psychedelic treatment or just a psychedelic experience, and then seeing how different sociological factors feed into their experience, feed into the positivity of it, the negativity of it, see what predicts treatment response, basically. I think that would be an interesting thing to do.
Well, yeah, I guess if you remove ethical considerations, the sky's the limit. You can
Yes,
ask any question.
It's a big if.
All right. Okay, Victor.
If money wasn't a problem, I think I would like to look at the pharmacodynamic differences in psychedelics in both the real humans and also AI systems and networks. So in that way, we can refine the AI system and see how it works. But at the same time, having this information and applying this experiment hundreds of times, and if not thousands of times, if you're running it that many times, maybe you can find a better insight on how the brain actually works of course, from our own concepts and biases. And maybe with that, which I think it can be some sort of an ethical issue here. It will be to create an artificial brain in the future, and that will be just out of pure curiosity, because I really don't see, as of now, I'll be hesitant to say how many benefits we would get as a society from having a real / artificial brain at work.
Yeah, I can foresee that happening. I wouldn't be surprised. All right, and finally, Thomas.
Hey, I've got an answer now. So two things that I've been thinking about a lot are big data in clinical psychology and better comparative studies between drugs. So if sky is the limit, I'd run like a 3 or 4 way comparison trial between like true empty sugar pill placebo, like amphetamine, MDMA, and 5-MAPB, and like cover a spectrum of like different kinds of stimulants in a talk therapy setting. Run however many hundreds of people through the system, get decent sample sizes, use AI to get all their textual data. You can video record the sessions and then do text to speech, and then use large language models to condense the themes. And I'd be really interested to see both the clinical outcomes and different thematic tendencies between each of these like drug therapy groups. That's what I'd do.
Cool. Awesome. Love it. Okay, well, thank you all so much for being here. Thank you all for the lively discussion. Thank you to our audience members for participating. Congrats to you guys for completing your talks. And that will complete the conference for today. Tomorrow will be day 3, which is our psychology and clinical applications theme day. We'll start off at 11 a.m. Eastern or 3 p.m. GMT. And we will see talks from Uma Chatterjee, from Dr. Roman Politsky, and from MAPS's Stephen Wong. So thank you all again and have a good night. See you tomorrow.
Thank you.